The neurons, which control our perceptions of the world, our thoughts, our drives, our emotions, our memories, these cells are essentially doing two things. They are conducting electrical messages along their lengths in an action potential, and where cells contact each other, then there are chemical signals which typically pass from one cell to the next in what's known as synaptic transmission, which will be the topic of this video. Now, there are some synapses in both neurons and muscle where electrical impulses can spread directly from a cell to a cell. But for the most part, that is not the case. So for the most part, where a cell contacts another cell, even, for example, here, if there's an electrical message which comes down the nerve cell and an electrical message which travels over the muscle cell, nevertheless, it has to be a chemical message which goes from the first cell to the second. Now, just to define some terminology, first, this is a synapse where a neuron is contacting another cell. Some synapses are between two neurons, so we could call it an interneuronal junction. This is a junction between a nerve cell and a muscle cell, so we could call it a neuromuscular junction. Then there are neuroglandular junctions where a neuron contacts a gland. And then there are more interesting junctions very often in the autonomic nervous system, or where immune cells are near neurons, and immune cells and nerve cells are talking to each other. But let's, for the purpose of this video, concentrate on neuromuscular junctions. And so we could come up with some names. So for example, this is the synapse where the two cells meet. And there will be a small space known as the synaptic cleft here. This first cell, the neuron, that's the cell that comes before the synapse. And so that could be called the presynaptic cell. This muscle cell is the cell that's coming after the synapse, and that is the postsynaptic cell. And so at a synapse, a message is going to travel from the presynaptic cell, this neuron, to the postsynaptic cell, which in this case, in the case of a neuromuscular junction, is a skeletal muscle cell. There may be a specialized area here, say in the muscle cell. There could be an area known as the motor end plate, where the muscle cell contacts the neuron, and the neuronal message could then cause a response in the postsynaptic cell, such as the contraction of a muscle, the secretion of hormone from a gland, etc. If you were to look at skeletal muscle tissue under the microscope, we can see these axons then reaching the cell. And I'm sorry, before I get to that. So once again, here in an interneuronal junction, this neuron, for example, all these little ones are going to contact this larger one. And so here is a synapse where they contact. This would then be the presynaptic neuron, and this is then the postsynaptic neuron. And as we'll see, you can have tens of thousands of synaptic knobs contacting this postsynaptic neuron. So, there can be a lot of synaptic activity here. And so here there are two neurons. As we will be discussing, there is a cleft, a synaptic cleft, where the presynaptic neuron, as we'll see, is going to be sending signals to the postsynaptic cell, and the postsynaptic cell will be waiting for it. So it knows that these chemical signals are coming, so that it would have receptors for those signals. Here you can see skeletal muscle fibers. They're the reddish elongated cells. And the darker lines are the nerve cells, which are then going to contact them so that synaptic knobs of the neuron would then contact these skeletal muscle cells. You can see the striations there. And this is where they would release their chemical signals, neurotransmitters, neuropeptides, and the like. And so this is based on contact. A nerve cell can only affect those muscle cells that it touches. You may recall the term motor unit from the chapter on muscles. So this neuron touches all of these muscle cells. They are its motor unit. So when that neuron goes to action potential, all of those muscle cells will contract. And so neurons can only influence those cells which they are directly contacting and releasing chemical signals there. Okay. So to go through this a little more slowly, action potentials travel the length of a cell as the membranes depolarize, sodium ions come rushing in, then the voltage regulated potassium gates open and potassium goes rushing out, and then the area returns to normal, and then that affects the next area, and this wave of electrical exchange passes over the excitable cell membrane of the axon, the axolemma. When it reaches the synaptic knob, there is another type of ion channel there, which we did not discuss earlier. It is a calcium channel. And so calcium channels then open, and then this brings calcium into the synaptic knob as a result of the action potential reaching here. Now, calcium is a very important ion inside cells used in second messenger systems. So, and this will become clear with hormones as well. When calcium comes into a cell, things happen. In this case, this synaptic knob has millions of these little blue bags called vesicles, each of which has thousands of neurotransmitter molecules. And so a handful of them will undergo exocytosis when the calcium is let into the synaptic knob. So the entrance of calcium will cause the exocytosis of the vesicles of neurotransmitter. The neurotransmitter will then cross this synaptic cleft and bind to receptors on the postsynaptic membrane. The cell here knows that neurotransmitter is coming, and so it makes the appropriate receptor. When neurotransmitters bind to the receptors here, they open ion gates. You may recall, and I'll go back to the video in a second, that some ion gates are ligand regulated, that they are closed when a neurotransmitter isn't present, but they open when the neurotransmitter is present. And once the ions once the ion gates open, then this muscle cell will then also depolarize to threshold. So nerve cells, neurons, are excitable. They can depolarize to threshold and start an electrical message, but so too can muscle cells. This muscle cell will depolarize to threshold. The action potential will then pass along the excitable cell membrane of the muscle cell, the sarcolemma, then through those T tubules to the SR where calcium is released. And as you may recall, and I'll review at the end, that this would then be the signal for the contraction of the muscle. Okay. So that was a quick overview, but now I'd like to go back. And so the neuron must then make the neurotransmitters, these signals. They are packaged into vesicles. And once again, a synaptic knob can have millions of vesicles, each of which then has thousands of molecules of things like dopamine or serotonin or acetylcholine, etc. Now, it is possible to make multiple signaling molecules. And in addition to things known as the official neurotransmitters, there are also other things like neuropeptides and neuromodulators, which also have influences at synapses. Perhaps not causing an action potential, but influencing the rate at which action potentials would be generated in the next cell. So when the action potential reaches the synaptic knob of the neuron, which is the presynaptic cell, calcium comes in. This will then promote the exocytosis of these vesicles of neurotransmitter, and this will then pump neurotransmitter molecules into the space that separates the two cells, that synaptic cleft. Once again, when we ask why are ions not free to move across the cell membrane, it's because these charged ions cannot cross this region where the nonpolar tails are located. But there are ion gates. Some, known as passive channels, are open all the time. And then others are open under certain conditions. So for example, some only open when the voltage changes during an action potential. This is a voltage regulated ion channel. But then others will open only when a certain chemical is present, like acetylcholine. So acetylcholine is a neurotransmitter, which is a signal for skeletal muscles to contract. So a neuron, which wants to tell a skeletal muscle to contract, releases acetylcholine from its vesicles. Acetylcholine crosses the synaptic cleft. And when acetylcholine then binds to the receptor, it causes it now to open its ion gate so that now sodium can enter the muscle cell, and the muscle cell can then depolarize. Now, this is a ligand-gated channel. It will remain open as long as the ligand, acetylcholine in this case, is present. But synapses also have an enzyme known as acetylcholine esterase. Acetylcholine esterase will break acetylcholine into its two components, acetate and choline. And then once that signal has been broken, then this ion gate will return to its resting conformation and will close again. And so two things happened here, both how to send a chemical signal and how to stop sending a chemical signal. So let me review both of those again. So if we want to send a chemical signal from this cell, the presynaptic neuron, to this cell, the postsynaptic cell, then calcium comes into the synaptic knob when an action potential reaches this point. This calcium causes the exocytosis of the vesicles of neurotransmitters. The neurotransmitters then cross the synaptic cleft and bind to receptors. This then causes ions to enter the postsynaptic cell, which could be a neuron, it could be a muscle cell. And this could then, in the case of muscle, start an action potential, or it could depolarize a glandular cell and influence then its secretion. So that's how the chemical signal is sent. If we want to stop sending the signal, all right, which is important because obviously I can tell a muscle to contract, but at some point I want to stop that message. I don't want it to contract forever. Then two things caused this message. One was calcium came in to promote exocytosis. And secondly, neurotransmitters were bound to the receptors. So those are then two things we have to stop if we want then the signal to stop. And the signal was not meant to last forever. So once calcium comes in, then there are calcium pumps, which will pump the calcium back out again. And once calcium is pumped out of the synaptic knob, this will end the exocytosis of the vesicles of neurotransmitter. And then in the case of acetylcholine, there is an enzyme which breaks up the neurotransmitter. If acetylcholine caused the ion gate to open, then if we break up acetylcholine, then that will cause the ion gate to close. So that enzyme acetylcholinesterase breaks up acetylcholine. Now, as I will pursue later when we discuss personality, there are different mechanisms for different neurotransmitters. So neurotransmitters can be broken up. They can also then be reabsorbed. So for example, here with serotonin, it is this neuron, which the presynaptic neuron, which is making the message, the neurotransmitter. And although serotonin can be degraded, there are enzymes which can break it up. There are also then pumps which can reabsorb the serotonin, so reuptake transporters. And so once again, we can not have a message last forever. Enzymes can break it down. It could simply, be washed away by, say, cerebrospinal fluid, but then also it could be taken up again through these transporters. Okay. And so there are different ways then of ending the messages. So we can both send chemical messages which start, and then we can also end those chemical messages. Just to return to skeletal muscle for a second, it is thus the electrical activity of the nerve cell, which then crosses the synapse as a chemical message, which then causes electrical activity in the muscle cell, which will result in the contraction of the muscle. We could call this the excitation-contraction coupling. As you may recall from the chapter on muscle tissue, once the ion gates at the synapse on the muscle cell open, then the muscle cell will depolarize to threshold. And action potential then travels over the cell membrane of the muscle cell, the sarcolemma. It then passes through the T tubules, which go from the exterior of the cell deep into the sarcoplasm. And then when it reaches that membrane system, the sarcoplasmic reticulum, the SR, there are voltage-regulated calcium gates which release calcium. If you recall, when calcium is released, things happen. And then, as we know, it is the calcium which then will start that sliding filament theory or process. So one more time, inside the muscle cell, once again, just a quick review of that idea from the muscle tissue chapter. There are these sarcomeres, and the sarcomeres have these proteins which will then pull on each other. So the myosin thick filaments pull on the actin thin filaments, and that's what will cause the contraction of the muscle. But at rest, the actin filaments are not accessible because they're covered by molecules like troponin and tropomyosin. But when the electrical message of the nerve cell crosses the synapse as a chemical neurotransmitter, it starts that electrical message traveling over the muscle cell membrane, the sarcolemma. This then goes through the T tubules, down, so it goes from the exterior of the cell through the T tubules to the sarcoplasmic reticulum, which then releases calcium. Calcium then binds to the troponin and moves tropomyosin out of the way. And so thus it was the nerve cell and its electrical activity, which became a chemical message at the synapse, which then resulted in the message which caused the contraction of the muscle cell. So this was that excitation-contraction coupling. Now, these topics are not only extremely important in just general anatomy and physiology functioning, but then when we look at the brain and personality, which I'll get into much later, I have a couple of playlists because, say, neurotransmitters like dopamine, they have to be made, and they're made in the presynaptic cell. But there are variations. Not everyone makes the same amount of dopamine, and variations in dopamine synthesis can be linked to different disorders, mood changes, and the like. Once dopamine is made in the presynaptic cell, it has to be it has to be perceived by the postsynaptic cell. And so there are receptors on the muscle cell, the neuron, whatever the second cell is. Well, these receptors, like dopamine receptor D2, these vary. They vary from one person to another, and their variations can then be significant for mood disorders, common differences in personality. What makes your personality different from mine? The variation in our dopamine receptors. And other receptors can play a role in this. Once dopamine is perceived, it could be broken down by enzymes. And as you might expect, there are variations from person to person in how we break down dopamine. And these can have consequences with personality, mood disorders, etc. Dopamine can be broken down. It can be taken up again as well in a dopamine transporter, and there are variations in that. So acetylcholine is an important neurotransmitter. Dopamine is an important neurotransmitter. Serotonin is an important neurotransmitter. And once again, when I talk about personality, once again later, a lot of this synaptic activity becomes relevant because the amount of neurotransmitter made in the presynaptic cells, the perception at receptors in the postsynaptic cells, the breakdown, the reuptake, all of these are important. And a lot of pharmaceutical products meant to affect mood are acting on synapses. A number of illegal drugs meant to affect mood are acting on synapses. A number of poisons act on synapses. And so therefore, this is very important for the topics in the nervous system, which we return to. Two last comments is this big neuron, the postsynaptic neuron, it has to decide whether or not to go to action potential or not. But if you look at who's giving it information, there are lots and lots of neurons with thousands and thousands of synaptic knobs. It can be more than 10,000 synaptic knobs contacting this one neuron. Some might be releasing one neurotransmitter, which is causing depolarization. So if one of these releases a neurotransmitter, which is getting this one closer to action potential, it's depolarizing the postsynaptic neuron. Its action potential becomes less negative. That's what's called an excitatory postsynaptic potential, an EPSP. So some of these signals may be exciting the postsynaptic neuron. But then there are also inhibitory neurotransmitters. So while some neurotransmitters like acetylcholine and glutamate tend to excite the postsynaptic cell, there are other ones like serotonin or dopamine or glycine or GABA, which tend to inhibit the postsynaptic neuron. And so some of these are sending messages which are hyperpolarizing the postsynaptic cell, making it more negative and less likely to go to action potential. So they are producing what are called inhibitory postsynaptic potentials, or IPSPs. So this postsynaptic neuron has to make a decision. Do I go to action potential or not? but it's got lots of information coming in, and some of that can even conflict. So the postsynaptic neuron is going to do two types of what's called summation. It's going to add together all of the effects of the EPSPs, which are depolarizing it and making it closer to action potential, and the IPSPs, which are inhibiting it, taking it farther from action potential. It adds all of those together. And there is an area called the trigger zone, which in this neuron would be at the base of the hillock, or I'm sorry, the base of the axon, which is known as the axon hillock. And if when you add all of these together, a little depolarization over here, a little hyperpolarization over there, what is the effect on the trigger zone? Does the trigger zone then go depolarize to the threshold voltage? If the answer is yes, then the postsynaptic cell starts an action potential. If the answer is no, then it doesn't. So this trigger zone decides to either go to an action potential or no, but it has to add together all of the EPSPs and the IPSPs from all of these diverse presynaptic neurons coming in. And because there is a specific area where a decision is made, all of these other synaptic knobs, it turns out their position matters, because the closer they are to the trigger zone, the greater the effect that they have. And so how does this postsynaptic neuron decide when to go to action potential or not? Well, it performs what's called spatial summation, adding together all of these EPSPs and IPSPs, and the position in space matters. The closer you are to the trigger zone, the greater the impact you have on whether the postsynaptic cell goes to action potential or not. However, there is also a type of summation known as temporal summation, because if this cell is close to the trigger zone and is sending the command, don't go to action potential, don't go to action potential, and this neuron is close to the trigger zone and is sending the message, don't go to action potential, don't go to action potential. But this neuron is excited. Excited neurons can fire more rapidly, and it could be sending the message, in essence, go, 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 go. And if that's the case, then this neuron, despite its position being farther from the trigger zone, could have a greater effect. So this postsynaptic neuron doesn't just add together the messages spatially, but also temporally. It pays attention to how quickly they come in. And an excited neuron, which is firing more rapidly, will thus have a greater effect. And so these nerve cells, these neurons, which are the base of our personality, our memories, our moods, our decisions, all of this, essentially they're doing two things. They're allowing electrical messages to pass along their axons, these action potentials, and also they communicate chemically at synapses. And so this video has been an overview of that synaptic transmission.